Occupational Health. Very glad that you came out today for our esteemed presenter, Dr. Christopher Morris. Chris is here from the Department of Global Health. Joined GW, how long ago was it?
8 months, I'll say.
8 months, fresh on the job. He has a really interesting background in terms of training, scientific training. He actually got his undergraduate Bachelor of Science degree from Rutgers University and Master of Science in Tropical Public Health and Doctor of Science in Immunology and Infectious Diseases from Harvard University at the School of Public Health there. He's been thinking long and hard about insects and mosquitoes in particular for a long time. He's studied all sorts of vector-borne diseases everywhere from chikungunya to dengue to Zika. And today, well, he's directing our Biological Safety Laboratory Level 3 for the School of Public Health, and right over at the Science and Engineering Hall. And hopefully he'll tell us a bit about what's happening there and what's in store, give us a preview, and also tell us more about your own current research interests.
Okay.
Welcome.
I'll not give this talk down. Thank you, Melissa. Thank you very much. Well, thanks everybody for having me. Happy to give this talk for EOH or get some neighbors down the hall if I live here. This talk it's about mosquitoes, and it kind of brings together a couple of different avenues of research that I've been engaged in for many years now. And so hopefully there's a little something in it for everybody. I think it's something we can all connect to. So everybody gets forged upon by mosquitoes, and everybody thinks they know everything they need to know about mosquitoes, and that they're horrible creatures and they ought to be done away with because there's just no real value to exist. And so we need to appreciate I think through this talk here, hopefully some of the data, the complexity of these systems and the lengths to which they'll go for evolutionary success. And then if you take the viruses that I study and put them on top of this system, you get some really cool stuff to study. And I guess I dedicate myself to it. But I think looking at this two-animal system, vectors and animal, the vector mosquito, and we'll just go with us, with humans for this talk. And then you put a virus that wants to live in both of us inside of the system there. The way that the pathogen can kind of play us off each other and use us to its best advantage is something really interesting. So I'll try and follow this outline. We'll see how far we get through it. I'll talk to us very quickly about what mosquitoes like, why do we care, what is mosquito exposure at a couple different levels, and then let's go ahead and add some infection to that mosquito and what it does to transmission. So why do mosquitoes bite? And which one of the mosquitoes bite? Everybody should know this, right? I mean, it's they need the protein from our blood cells to be able to break that down into amino acids and then use it for the building blocks of the telogenin, which is their egg yolk protein that they then send to their ovaries to make little mosquitoes to offend us further. So it's purely a protein source for them, really. They can get some energy out of it, but really they get their energy from feeding on plants. So they're nectar feeders. I suppose they could be pollinators, but how important that is in terms of their role on Earth is probably nothing anybody wants to know. But that's why they're taken in. So only the females, of course, take blood meals. The males, they're just kind of there for fun, and they do their job and then go off and I guess consider only pollination. So here's a female mosquito, and this is one of the first things we need to confront is that oftentimes they're thought to be just flying syringes, abiotic processes in a model. They're forgiven a lot of the details in their infected life cycles and infection dynamics, and so this is not true, right? Mosquitoes have a whole lot under the hood, and the viruses and parasites that infect them don't really treat them as sort of mechanical deliverers of the infectious nodule. When we talk about the business end of the stick here, okay, and a mosquito and the mouth parts, you see this big proboscis, right? But really that's just a fleshy sheath that covers this really cool armamentarium of 6 to distinct pieces of feeding parts. So you have maxillae, which saw through your skin, saw through the epidermis, so you can get a hole in there so you get down to the blood that they want. You have the mandibles that are like tongs on your barbecue and kind of like push and pull meat out of the way, right? So it can go ahead and insert then its labrum and its hypothalamus. And so the labrum is actually the tube that's going to go and suck your blood up through, and the hypothalamus is where you're going to have the delivery of salivary products, which it uses to sense and then disrupt hemostasis so it can actually feed the depletion. So, you know, it's quite a lot of tools it brings to the picture. This is something that I think is just really cool to show. It's a video of, a transdermal video of the probing action, right? So most of us think it's just, again, that big thing going in and just cannulates a vessel and it's done. This is a nice video that was done by a group to show how flexible this tool is. So you can see the proboscis really bending around inside of the tissue here in the mouse, looking, poking holes in everything it touches, spitting all the time, trying to sense for blood products, adenosine, triphosphate is one of the main indicators that it's hit something. And so here you can see getting closer to a vessel now, and bang, it cannulates. And so that's a good feed. Okay. So it's not a syringe, like, you know, it's really a very cool tool. And so this is cannulation, okay? And another way it does it though is like while it's probing around there, it just nicks a vessel, right? Or strips some capillaries up, and you get a hematoma there in the dermis, and it'll stop on that too and just feed out a pool of blood that collects there as well. So you can get a couple different things. This, you know, trying to tell you that there's a, we have, significant p-values in terms of detecting whether or not something's cannulated or pool fed in terms of any sort of untoward outcome. I can't suggest to you, but they don't always hit their mark like this. Okay, so this is all cool. Why do we care about mosquitoes though? Well, we care when our bodies release histamine and create itchy welts and things like this. And so this happens when degranulating mast cells that are resident in your dermis that have surface-bound IgE see some of that spit, see some of those salivary proteins that the mosquitoes injecting into you, binds up the AIG, degranulates the mast cell, and then it's like confetti everywhere. The party starts because the mast cell is going to release all kinds of mediators to essentially attack whatever the problem is at that location. This is a blunt instrument. It's not really something very specific, and it's going to call in all the forces to say, hey, there's something going on here. You better come check it out. Immune system, and it could be something we need T cell responses for. It could be something that there's antibodies for. Innate and adaptive responses will be called in following the mast cell degranulation. But really, we care because it creates an itchy mess in the well. Then we scratch it, it gets infected. We also might care because, at least here in this building, we care about disease. And of course, mosquitoes are just, most deadly animal on Earth and all that stuff. And so, everybody likes to say, I love this colorful blocking WHO is nice. Just again, trying to call attention that, mosquitoes do still kill a heck of a lot of people, especially because of malaria, especially in Africa. But things are definitely on the move, and Aedes aegypti, now is endemic for about half the world's population. We've seen fantastic new outbreaks of Aedes aegypti driven viruses, especially here in the Western Hemisphere. And to kind of to get away from some of those outbreak phenomenon, the thing about dengue having increased 30-fold over the past 3 decades, that really is something quite remarkable. I mean, when I started my career off, dengue was really rather neglected. It was just there's a fever in Southeast Asia, but it's really changed its transmission dynamics. So, talking about mosquito exposures on a population level now, which is one important way that we study these things. We know that, of course, mosquitoes exist. People get bitten. Sometimes you get infection. What we don't appreciate well, though, in these systems, well, there's a whole bunch of things we don't appreciate. our specific contact with mosquitoes. The result of this, meaning we don't know kind of where, when, or how many of these things we're going to interact with at a population level, is that most of our interventions against mosquito-borne diseases are much less effective than we would desire. Okay? We're pretty good at detecting disease in ourselves, even disease in mosquito populations, even though we don't really invest much in that. But when it comes time to say, like, wow, this looks like an outbreak, somebody go in there and do something about it, we find that our tool shed is quite limited in terms of what we have available to issue that phenomenon. So, one of the ways we realize this and we explore transmission is through mathematical models, population level models. These are some of the ways that we traditionally look at vector-borne diseases, originally created for malaria, but been adapted for all kinds of other things. You'll recognize here the Ross-McDonald model for R0, looking at vectorial capacity times essentially vertebrate efficiency, or the recovery rate times the intensity of the infection. Vectorial capacity is an entomologic representation of the vector population that's taking up the infection and passing it along. And I'm not going to go through all this, but just to call out the one term here that is contact-based, really. So A is the biting rate, okay? In this case, we'll call it the man biting rate. It's raised to the square because essentially it has to bite twice to pick up and then transmit, because we're talking about non-heritable infections, okay? And it's really not a very cool term. It doesn't have a lot of mathematical power to it. I mean, it's more than abundance, okay, which is M. But it's something that's not intellectually really exciting. So it gets somewhat neglected. And so we can do all kinds of other fantastic models and experimental efforts to explore. In this case, this was a bunch of different dengue strains, and we looked at in a mouse model and in vector models, this is in vectors here, the differences in titer based on different strains of dengue over time. And we can come up with all kinds of fun mathematical representations of just how much more efficient or less efficient strains are than each other, to come up with, like, this is the best dengue we've ever seen, or this is a weak one. It's going to have less of an outbreak potential. In animals, we can do the same thing. We can create these animal transmission models or infection models, where we're able to learn all kinds of things about these pathogens. Again, using the mosquito as an inoculator or initiator of the infection. And this gives us a look into what happens then after a vector-borne infection and the course of viremia, the size of the window, the peak of viremia, all of which then relates back to how infectious they probably are to the next mosquito bite. We can complete that cycle too. But this and the previous slide kind of lives within the animal, right? The previous one lives within the vector, all about extrinsic incubation period. Here, the intrinsic incubation period. And we don't talk much about this thing right here, where they actually touch, right? And when and where they touch is really quite critical, because all these other infection dynamic processes have some relevance to when that blood meal is taken, right? And so at a minimum. And so here's a viremia curve, okay? And what you can see is that if you're a mosquito and you're coming in, you're going to feed on this viremic animal, the day you get to that animal is going to have make a big difference in just how much infectious inoculum you are exposed to, and then what your potential for infection for yourself is, right? Not that they care about that, of course, but it's what it does is it shows you that it's not as simple as a mosquito binding into a viremic animal and it's purely Brownian and everybody who touches gets infected. Okay, which unfortunately a lot of models do, and a lot of thinking that goes into policy does as well. So transmission models can be used to measure things like A, or at least like Q here, which is an acquisition of infection model, where we can model essentially or measure like different levels of viremia and what those outcomes are. This can be done also in humans, and so we do this with naturally infected people and feeding mosquitoes on them in the field as well, to derive better parameter space for some of these things. using all this stuff, we can, and vector density collections and stuff like this, we can come up with exposure risk maps. Okay, this is a little bit better. We're getting some information about the variability over space and time as to when and where seasonally and spatially you're more likely to come into contact with a vector population at a minimum, but really we're mostly talking about vector populations that have some level of exposure to them. We can run those through the models and come up with risk maps as well, and infer the risk of emergence due to introduction to these areas because of underlying ecological susceptibilities or populations. We can do this then by looking at, if you think about Zika, if you think about chikungunya, these introductions to this totally naive hemisphere and the trajectory they took, this becomes kind of interesting to think about. But also, in the years before that, if you look at things like Miami or southern Florida or southern Texas, where they have annual introductions of dengue, and sometimes they take off, not much, but they might do more than just cause, 0 to 1 autochthonous cases. A bad year would be 10, 15. But you can look here then at, okay, well, given an incoming traveler so many days into their infection and a waning viremia most likely, and then varying contact rate or how many bites per day someone, that person kind of experiences, we can put that into some of these models and come up with an idea about what the probability of more than one autochthonous case would be. So how likely it is that you get secondary and tertiary cases. This is another way of looking at it here, where we're changing the biting rate from a whole lot of biting going on, right? 0.75 bites a day down to 0.25 bites a day, and what you see is that this is all pretty clean, but when you get down here to a lower bite rate, you can see that the model doesn't coalesce as nicely around an obvious outbreak prediction, and so you can see again that biting rate itself can destabilize some of these predictions, and that's really where we need to probably focus more of our attention and say, well, is this a realistic biting rate? Just because we get a cool outbreak every time we put it in, and we're all citing some study that happened 20 years ago, because biting rates are, they're derived strangely, and sometimes it's really difficult to get IRB to approve landing rates again. Not that those are great either, but there's a lot of pushback on how you actually measure the bite rate, right? So this sort of contact between mosquito populations and human populations is absolutely necessary for transmission to occur. I hope we all agree, but it's not sufficient because there's so many other parameters inside of those models, and so much else going on inside of both animals there and their interaction that really flavors this contact in terms of the transmission efficiency or viral efficiency afterwards. And so once that contact happens, what I'm going to show you now is that another battle begins here between us and the mosquitoes, and this is about exposure at more or less an individual level, which of course we can still extrapolate, but this is going to be really talking about what happens inside of a singly foraged upon individual. So the probing that accompanies the deposition of mosquito salivary proteins, these proteins, include really powerful vasoactive, anticoagulative, analgesic proteins that enhance the mosquito's blood feeding success. There's no virus in this part of the talk. We're just talking about empty mosquitoes having a good time, getting their egg yolk protein. This little picture here is a 2D gel of mosquito saliva from Aedes aegypti. So we sized it and put it over a pH gradient of 3 to 10 to isolate individual mosquito salivary proteins. This is from a pool of mosquitoes. You can't get that much protein out of any one mosquito, of course. And maybe I look like a dinosaur because I'm using a 2D gel, but I'm a visual person. So, but through this process, what we were able to do is we were able to, confirm some prior findings about what kind of proteins were in there. Certainly, it launched into a bunch of other investigations. I'll show you. It's not a couple of things. There's well over 100 distinct proteins inside of this cocktail that's being inoculated inside of you with every probing event, and a lot of them are quite powerful, quite antigenic, and so we'll show you some of the details there. Some key ones that people have studied, that we've studied, inside of Aedes of saliva, okay, include sialokinin, which is causes vasodilation, adenosine deaminase, which suppresses pain signaling, okay. It also helps prevent mast cell degranulation by breaking down adenosine at the site. Epirase and Egyptin, which work together along with a number of others to stop coagulation, to keep the blood flowing essentially, right? Enhance the feeding success. Serum proteases also, and the serpines to again try and enhance blood flow to the bite site. When we talk about arthropod saliva, some of these things are acting in terms of anti-hemostatics, okay? And so some of them, like the sialokinins, are going to help to dilate the vessels instead of constrict them. Some are going to prevent platelet aggregation. Some are going to prevent complement fixation. And so anti-hemostasis is a sort of a natural, that makes sense. Mosquito needs to feed. Our body's trying to plug the hole, so it's going to inject things that help it get its blood meal out. But also a lot of these things are immunomodulatory. And so they try and damp down inflammation or redirect inflammation to certain pathways to reduce cytokines that might be disruptive to the mosquito's feeding behavior at that moment. And all this has to be put in the context of the really limited temporality that the mosquito is trying to get its blood meal. We're talking about maximally 10 minutes, but really mosquitoes kind of get it done in a few seconds if they cannulate the vessel like they should in that video, okay? So these are really quick processes. There's not a whole lot of time for our bodies to necessarily respond with more than what we have armed there in the dermis already, okay? So, in terms of the immunomodulatory components of saliva, it's important to realize that a whole lot of the dermis is part of the immune system. It has immune function. The epidermis itself is avascular, but there is some suggestion that even that Langerhans cells and keratinocytes can be productively infected. But the dermis is really where most research is focused, and where a lot of the movement of virus in and out in cellular components seems to take place. Indeed, for dengue at least, these are some prime targets for infection, even though they're resident, responding, and mobile, and oftentimes mobile cells that are going to take virus away from the bite site in a way that kind of protects it from whatever sort of toxic response is going on there during the inflammatory response to the bite itself. So, in a nutshell, what are we trying to do if we're Aedes aegypti and spitting our saliva in there? We're trying to inhibit inflammation. We're trying to suppress a whole lot of Th1 style responses, and try and tell the body, no, don't worry about us. Go ahead and recruit some antibody instead. It's going to be much better for you. We'll be out of here by then anyway. We're going to also, but part of it is to try and say, we need this vessel that we're in, or that we're trying to keep open and bleeding, to relax and to let more blood come in. And so that's going to bring in more neutrophils and eosinophils, but probably the mosquito's getting out of the way by the time that's fully realized. So, in terms of immunity, a lot of these proteins that I showed you on that 2D gel are going to be found in here. Some of them are going to help reduce degranulation of mast cells, suppress prostaglandins like TNF-alpha, suppress the salps and such. They can suppress the complement system. And then for the adaptive side of the house, they really damp down this Th1 response, pushing things through IL-4 and IL-10 to a Th2 response, which again seems to favor the blood feeding success. So yeah, no virus in here at all yet. This is just mosquito feeding. So it has many biologically active components. We see these components. Okay, so a lot of them are antigens, very strong. Some are very strong antigens, and so we can produce immune responses to these proteins. And so if you figure, you only get one mosquito bite, one first mosquito bite ever, and then every one after that is just a booster, right? We can measure in people through all sorts of antibody and antibody subclass exposure to specific mosquito salivary antigens or total antigens. And there's a good bit of activity right now in trying to tease that apart and find a clean way to look at this in terms of mosquito contact. So getting back to, we don't really know how to measure contact. We don't have a good way to do it. This is one way people are trying to get at this, is say maybe there's a serial marker for how many mosquito bites you're getting, how heavily forage applying you are. And so that's an active space for MyLab and a number of others. So one of the things we found when we looked at Aedes aegypti in a seasonal way, exposures in people, which was really kind of encouraging and interesting, is that we found that people would have a seasonal bump in their IgG titers, okay, that would wane really quickly. So essentially what we did in this case was we went to Norte de Santander in Colombia, and we set up a study site up in Pamplona, which is above the Aedes aegypti line, about 1,600 meters up, I think we are. University there, all the kids come from Cúcuta, which is down much closer to sea level, lots of dengue, lots of transmission. No aegypti up at the university site, and so we enroll them when they show up to classes at the start of the term, get measure their antibodies, let them track them over the course of the season, and they go back home, come back, and we'd be able to see a bump in their titer when they came back to campus and a decay over the course of the semester when they were up there. So these are pretty dynamic. I mean, you think about IgG, and you're like, well, it's forever, right? I mean, your IgG is going to, well, more or less forever. It shouldn't be this time, right? But there's something to the exposure to these salivary proteins, how they're recognized, maybe how often they're recognized, that leads to this really very dynamic sort of response that we've seen in terms of our antibody response to them. So this might give room for this to become a tool for some sort of seasonal diagnosis of exposure at least. So, this again just kind of confirms it. It's another study we just had come out, looked at subclasses of IgG. So instead of total IgG here, we looked at IgG1, IgG4. IgG4 is a very strange antibody. I don't know if anybody knows anything about it, but it's a lower proportion populations, like about 5% of total IgG is IgG4. It can make these strange hybrid antibodies where it changes Fab fragments between itself and others. But it also seems to give us a pretty good dynamic response. And so we get a cleaner look at some of these seasonal differences or exposure differences that I'm telling you when we focus on IgG4. And so you see this peak response here is a really kind of acute antigen positive, PCR positive, and everybody's IgM and IgG positive in that case, response to mosquito salivary proteins in a total way for IgG4. This next bar here is they're no longer nucleic acid positive or antigen positive, and so it's the waning of that infectious process. And so there's again just a dynamism to it. Is what this sort of the idea is here, was to work towards trying to define an exposure metric for mosquitoes. So moving now into, let's add some virus to the mix. It's important to, again, remember that this is not a syringe. It has its own biological processes. Arboviruses are in the vector, okay? So this is an important host for them as well. They come in here through the blood meal, get into the midgut. They have a very short amount of time to escape this highly toxic environment, because what's going to happen within minutes is you're going to have the formation of a matrix called the peritrophic membrane, which encapsulates this bag of blood, squeezes all the water out of it, while it also dumps proteases in to break down all of that fresh blood into the building blocks of the gel. Viruses being non-motile, of course, we have to think about this in terms of now maybe something a little more fluid dynamic, or at least probability based in terms of the three-dimensional envelope of that midgut and the proximity of virus to the receptors on the midgut epithelium that can then take that virus out of that environment before it gets destroyed by the proteases and set it up for infection of not only the midgut epithelium, but then ultimately in a competent vector, it'll escape into the hemolymph. It's an open circulatory system, arthropod, so once it's out, it's everywhere. Sets up infection in the salivary glands, and then is within, depending on the ambient temperature, 3, 4, 5, 6, 7 days away from transmission. So, knowing that there's all these cool proteins in saliva, and the virus has got to be sitting in there, basking in them in the salivary glands, and doing something that, probably enjoying it to some extent, right? It's able to have some sort of impact. I submit, being the most plastic organism in the system, right? It has the ability to explore its own genetic potential much more readily than the mosquito does, or we do, to respond to it. I said, I bet you we can look at this saliva and we can see the impact of the virus in it, and that may just tell us some important proteins in there in the saliva for onward transmission or perhaps even pathogenesis. So again, when you've got a virus in the saliva, the probing vent that I showed you the video for, is going to simultaneously inject saliva and virus through the same duct there, as the proboscis is searching around for a blood pool or blood meal. Other people have looked at the effect of saliva on disease establishment. So in viral diseases, some friends of mine looked at the saliva increase in West Nile virus, Aedes aegypti, and it's also, the prototypical was the mexidillin in sandfly saliva has been shown to modulate leishmania establishment. There's also bacterial infections. This is for Borrelia burgdorferi, Sal-15, increased infection of ticks, and suppresses the immunity in the skin it's deposited into. Ticks, of course, are going to be a very different scenario. They're going to have to feed for a much longer period of time. They're going to have to, evade that immune response to a much greater extent than a very time-limited feeding like mosquito will. And then prior to our getting involved in it, one of my colleagues, Dan Calvin, studied mosquito salivary proteins in dengue virus. This was based on salivary gland extract that you'll see sometimes in the literature. That's when you take, people excise the entire salivary gland and just homogenize it and use it as an antigen prep to look at proteins or immune markers. The problem with that is that there's a whole lot of other stuff in there, like mosquito tissue, that's not true saliva, and so it can be a bit muddier sifting through SGE results, but it still has some utility, and it's certainly a lot easier than looking at mosquitoes for saliva. So as we went into this investigation of looking at what's the impact of virus on mosquito saliva here, we started with some transcriptional assays and moved on to the 2 protein work, and we were able to see transcriptionally that there's a whole bunch of things here that dengue is doing to mosquitoes' salivary glands and mosquito saliva, and some of that makes sense because the virus is it's taking a toll. It's taking you can see it's a lot of hits here in the mitochondrial and energy stores. The virus is taking over the machinery of the mosquito for its own replication. So a lot of that is, I think, what you're seeing when you look at salivary gland transcriptional assays and salivary gland protein expression. You're looking at the impact of the infection upon that organ. Okay. So that's when I said, okay, we gotta get beyond the structural organ and get just to the essence, the saliva itself. And so this again is a 2D gel, just you know, a different one where we ran the saliva out, and what we did was I did infected and uninfected salivary preps. Okay, and so then we were able to do some protein analysis and look at up and down blood regulation. You know, I of course predicted that we'd find a magical protein that was upregulated and give us all of the patterns and answers we needed.
Need a drum roll.
And and of course everything's kind of downregulated, but not everything, but a whole bunch of things are downregulated, and they're downregulated in very specific ways, okay, and dramatic ways. So a number of these proteins are just crushed, okay, as compared to an uninfected mosquito. When it comes to feed on you, this mosquito is going to go out that's got dengue, and it's going to be missing a whole bunch of its salivary tools, okay. So what's the impact of that? So looking at what these proteins are and what they mean for successful feeding and transmission, that's going to give us some clue as to what maybe what the virus needs to succeed. So some of these things that we found, some of the big ones like Egyptin, adenosine deaminase, okay, we can kind of figure out pretty quickly what they're about. So Egyptin binds collagen, prevents interaction with von Willebrand, inhibits platelet adhesion aggregation. Okay, so it's going to help keep blood flowing. It's going to prevent clotting. It's going to make it easy for that mosquito to get its blood meal. So why does the virus want to make it harder for that mosquito to take its blood meal? Adenosine deaminase suppresses pain signaling and mast cell degranulation. Okay, inhibits proinflammatory cytokine production. Why would a virus want to say, oh, it's okay, go ahead and degranulate mast cell, when that degranulated mast cell is going to make a wildly antiviral environment for the virus. So these are kind of counterintuitive in some ways. You just got to think about them. Okay, what would be the advantage of this? And some of it's this temporality again. We don't stick around there forever. What is the immediate need of that virus to get in and get out? We took to explore some of these individual proteins. We cloned and expressed them, made them recombinantly for single mosquito salivary protein injections that we put in along with dengue, so we can kind of see what the impact was of these individual proteins, besides our predictions. We did them in our mouse model, which is missing some interferon regulatory factors. It just kind of delays the onset of interferon and gives us room to see a viremia. So, but otherwise they're intact and they will clear the infection. So we gave injections in the ears, sometimes in the feet, but I think this study all use ears. And then we looked at dengue 2 viremias and cytokine panels. And so for single salivary protein injection, dengue 2 plus Egyptin, we're able to see AV is Egyptin and virus. Okay, so we're able to see much higher levels of interferon gamma. Again, something you don't think a virus would really want to see happening in there. And a whole lot of eosinophil infiltration, and a reduction in monocytes, which the dengue virus can really take advantage of in terms of a safe harbor, replicating thing. So that's kind of, again, it's like, okay, well, that doesn't make a lot of sense to us. And you see here, though, that with that same inoculation, we get decreased viral titers at the inoculation site. Okay, so okay, so maybe that's why it's taking the Egyptin out. Maybe it's because it enhance viral titers at the site. But some of this again is like we've become now wildly artificial just to make sure everybody's following this. Okay, we have now taken virus and inoculated it with a needle, and we've injected a single protein God knows what, I mean, we know concentrations, but God knows what, really physiologically relevant concentration is compared to what the mosquito puts in. And, it's not going at the exact same spot. So there's some artificiality here to this. But, you can use this as some sort of a starting point, at least have the conversation about, okay, well, why still would Egyptian be important to reduce in terms of its impact on an infected bite as compared to an uninfected bite? So again, just a decreased viral titers here. Adenosine deaminase, we saw the same thing. We saw decreased viral titers, not as significantly so, but again, this seems to go against what we would expect the virus would prefer to have this signal. But again, are we overloading it with these proteins? And so, we're seeing some artificial effect here as compared to what a naturally infected and reduced Aegypti saliva plus virus would do to these animals. When we take these findings, these reduced protein levels, and some of these individual protein experiments together, and we think about them in terms of transmission modification, okay, we can come up with some interesting model concepts that are worth exploring and contemplating. This is just some of the ways we think that the totality of this dysregulation of these proteins could be acting to enhance dengue. So by exposing the bite site with dengue to this ultrasalivary proteome, okay, we're going to perhaps make it more difficult for the mosquito to take a full blood meal. Okay, we're going to decrease these anticoagulation factors. We're going to make it so that maybe it's more difficult for the mosquito to fully feed or to find a blood vessel and keep it open. And so it might keep on expectorating, right, or probing in other locations and increase the viral inoculum. There's some literature to suggest that yes, the viral inoculum and how much it gets put in is related to infection success, and also pathogenesis, although I don't think the mosquito cares, or the virus cares about that too much. But this could result in this sort of attempted refeeding on the same host, increasing the inoculum. Okay. In terms of the adenosine deaminase stuff, okay, which is the pain suppression, right? What we think might be going on there is by increasing pain sensation in the host, and making it more difficult here to feed because of the hemostatic dysregulation or reconstitution, it might be more likely the host detects the feeding attempt, okay? And the mosquito is alighted, if not killed, to another host. Because it's not like other systems like maybe Lyme disease, something like that, or a bacterial pathogen, where you have a long reactivation period for the parasite inside the salivary glands. Here in the mosquito, the virus is ready to come out, the first little microliter of spit that comes out. So as soon as that mosquito spits its first bit of saliva in there, infection is likely going to ensue. So putting more in doesn't make a lot of sense. Making it difficult to finish and more dangerous to stay because you've increased pain sensation might cause it to alight and go to another host. And then for every time that mosquito goes to somebody else, it's then, the virus has doubled its transmission. Okay? So this is a way to kind of take these interesting protein findings and put them in the context of, well, okay, we didn't get a magical, proviral or antiviral bullet here, but in terms of contact, it kind of it can make some sense. And so, looking at these mosquitoes now, and trying to drill down instead of like individual protein expression like we were doing before, now the approach is to engineer out through CRISPR mechanism some of these proteins, so we can really just obliterate some of these individual proteins and start looking at them onesie-twosie in a modified mosquito background there, and see if we can make some of these measurements in terms of time to feeding repletion, time to alight. These are things we can then do experimentally to challenge this set of hypotheses. So all these mosquito salivary proteins, they give us some opportunities to perhaps develop interventions as well. As I said, I was hoping for a magical protein that we could patent and I could retire on. You know, of course I still want to work. So, but there's a whole bunch of things in here. I mean, if it's pro-viral in some way, the opposite of it is antiviral. And so, you know, I guess you could say a minimum, right? You could try and say if the virus prefers not to have that protein there, then putting it back might be a good thing. And so it might be that there's a way to create a therapeutic around it. Alternatively, if you're trying to deliver maybe a banged up virus in terms of vaccine formulation, and you know there's some adjuvanting effect of some of these proteins here, they could become interesting additions to a future vaccine formulation where you're trying to expose someone to a construct that's just not, they're not even thinking about what that immediate environment is like and what a virus might prefer to present itself to the cells. So you're looking at this as in terms of advanced adjuvant development and foraging room. It's a long-held idea. It used to be called bugs to drugs, and so there is interest in trying to find these things out here at the individual protein level. And so I also hit on I think diagnostics through some of the epi work there in Columbia. We're looking at exposure. Well, so as we break down this total IgG against mosquito salivary protein into individual assays against individual salivary proteins, and then dial that in further to just the IgG4 subclass, we can really start to consider how we might create an effective contact tracing serodiagnostic. There will be, of course, some substantial limits on this because it's still going to be based on your total exposure. We know that IgG responsiveness wanes over time, and so there is it looks like there's some tolerance over time, or else you just don't get foraged on as much as an adult because peak IgG4 levels are in the young children and teenage years, and then as you go into your 20s and 30s, it tails off. So, so it might be that that again, it needs to be tuned for either use in young populations or not as effective an indicator in older populations. But it's certainly something dynamic that should be used to bring to bear on the problem. So that's mosquito exposures in a nutshell. So we did mosquitoes on people and population-wide. We did mosquitoes empty, mosquitoes with virus, and I haven't had any questions. So.
Thank you for a really interesting talk. So how do mosquitoes choose their target? Everyone will tell you you're sitting with certain people. I always get bitten, I don't get bitten. What's the...
Yeah, I mean, there's just a whole lot of hearsay and a little bit of dogma in there, but none of it really true too well. And so it's people with ABO blood group, they tend to be bitten more according to one study. People who drink beer apparently get bitten more. Maybe they're just hanging out drunk without a shirt. I mean, people with stinky feet, but maybe it's because they're airing their feet out because they stink so bad and they get bitten more because they have their socks off. I mean, there's a whole bunch of reasons that things you can do to debunk all of these. I mean, we know that they're attracted to CO2. We know they're attracted to big heat. We know they're attracted to, some are attracted to octanol, which comes out of stinky feet too, stinky cheese. But there are some attractants. They don't transcend all